Good afternoon, everyone, and welcome to the EAS Stage. I'm Pelin Golforoush, and together with my colleague Tatyana Storozhenko, we are really excited to host this session today. When we put this panel together, we wanted to do something a little bit different. Rather than focus on one topic, we wanted to bring together three rising voices in our field who are all working on the same big question. How do we beat atherosclerosis? But coming at it from completely different angles.
Hello, everyone, and thank you, Pelin, for the introduction. So today we have three labs from three different countries. Dr. Soheil Saeedi from the University of Zurich, who leads the cardiovascular aging lab at the Center of Translation and Experimental Cardiology. And Dr. Saeedi works at the intersection of gut microbiome and cardiovascular aging. Dr. Cesare Canepari from the San Raffaele Telethon Institute for gene therapy, where he's working on using gene therapy and genome editing to cure FH. And Dr. Berkan Kurt from University Hospital Aachen. He's currently cardiology resident and clinician-scientist working on inflammation and cardiometabolic disease. Thank you so much for joining us today. And before we go deep, I would like to ask you to introduce yourself to our listeners, who you are behind the science.
Thank you very much for having me today. And thank you very much for the EAS for this recognition. It's truly an honor. My name is Dr. Soheil Saeedi, as you already introduced, the group leader and head of cardiovascular aging lab at the University of Zurich. My lab research focuses on the interconnection between gut microbiota and cardiovascular aging. And we in the lab try to identify novel microbial metabolites that causally contribute to cardiovascular aging and related diseases. We also characterize and formulate microbiome-based strategies to tackle cardiovascular aging and diseases.
Thank you for the invitation. I'm Cesare Canepari, and I'm a postdoc in San Raffaele in Milan. We focused on gene therapy, mainly to the liver, and we recently started working on hypercholesterolemia. So we published a work on LDLR gene transfer to the liver by lentiviral vectors to tackle the pediatric population of affected by the most severe form of hypercholesterolemia, which is the homozygous form. And more recently, we started a project on genome editing and particularly base editing to tackle more common forms also of dyslipidemia.
Thank you, Tatyana and Pelin, for organizing this. And I would also like to thank the EAS for this recognition. My name is Berkan Kurt. I'm a clinician-scientist and cardiology resident at the University Hospital in Aachen and work in the group of Professor Florian Kahles. And collaboratively, we work on the role of inflammation in cardiovascular disease and cardiometabolic disease. Since in the last decades, we have learned that especially inflammation is the link between risk factors and disease development. And we try to translate the findings that we have seen in the last decades to clinical routine practice. And I'm happy to talk about this later on. Thank you.
Thank you for those wonderful introductions. So now I'll go on to specific questions to each of you. We'll start with Soheil. Your lab works on cardiovascular aging, and one of your recent papers in Nature Aging showed that a gut microbial metabolite, phenylacetic acid, can actually drive endothelial cell senescence. Why does this matter, and how did you even end up looking there?
Thank you for the question. First of all, I want to indicate the important note that vascular senescence and aging are both early biosensors and drivers of whole body aging. Now, the critical question is why some people develop vascular aging phenotype and related diseases much faster than the others, even beyond classical risk factors. The answer to this enigma could be gut microbiota. And today we know that gut microbiota can produce and release of thousands of chemical compounds toward the host, which are circulating in the bloodstream and reach out blood vessels. In this project, which is partly published in Nature Aging, and another follow-up study is currently under revision in Cell Reports. We'll keep fingers crossed for that. In this project, we identified, as you already indicated, microbial metabolite phenylacetic acid and its derivative PAGL and phenylacetylglutamine increase with age and atherosclerosis. They can also predict 1-year MACE and mortality in aged patients with ACS and ASCVD. But the important and exciting part of the project is that they are not only predictive biomarkers. PA especially can also causally induce vascular niche senescence and drive atherosclerosis. Mechanistically, it induces epigenetic reprogramming, impairs reduction homeostasis, and boosts SASP as one of the hallmarks of senescence in the vascular endothelial cells and perivascular adipose tissue, reinforcing pro-inflammatory and pro-atherogenic microenvironment. The most important take-home message of this project is that gut microbiota is not only a bystander, is a modifiable target. We are currently working in another project, which is again follow-up study, to target a specific bacteria that produce this metabolite to slow vascular aging and prevent vascular diseases in aging population especially.
That's very exciting, and it's a very fascinating way to frame it. Thank you. One very interesting idea in your paper is the concept of microbiome-based xenotherapy. How realistic do you think it is that interventions targeting microbial metabolites could eventually become part of cardiovascular prevention strategies?
Important question. When we talk about xenotherapy, the first thing comes to our mind is senolytics, xenomorphics, the chemical and natural compounds, or even cells that directly eliminate senescent cells or suppress SASP and restore proliferation in senescent cells. But these are not the only xenotherapeutic compound that we can find outside our body. Still, we have such compounds inside our body which are produced by microbes in the intestine. You see that I'm talking about some xenotherapeutic compounds inside our body. It seems that there's a battle between good and bad bacteria, good and bad metabolites. We had PAA, PAGLN as the bad ones, and this time we have good one, which is anti-senescence compound. And that was acetate, which is a short-chain fatty acid produced by a specific bacteria like Bifidobacterium. And what we found is that this good metabolite can restore proliferation, mitochondrial metabolism, and suppresses SASP, and can act as a xenomorphic compound. So another take-home message from this project is that again, microbiota is a modifiable target, and if we can make a balance between the good and bad bacteria that produce such metabolites, we might be able to somehow regulate the vascular aging process and prevent vascular diseases.
So we're somewhere between concept and clinic. If you had to pick one thing in the field of cardiovascular aging that we aren't paying enough attention to, what would it be?
In my eyes, I think that the field still underestimates how interconnected aging process is. We often target aging-related molecular mechanisms in the heart or blood vessels and their cells, while aging is a systemic and multi-organ process. Based on very recent science paper, we know that aging is half and half genetics and epigenetics, and environmental exposure and its elements can somehow regulate epigenetic alterations in aging. In cardiovascular context, despite of aggressive and efficient management of classical risk factors, residual risk still persists, and more than 30% of all cardiovascular events occur. And what's behind? Again, environmental exposure. And one of the major elements in this exposure is gut microbiota. What we know is very limited still. in the context of cardiovascular disease, because the first paper that reported causal contribution of gut microbiota and its metabolites to cardiovascular disease returns to 15 years ago in 2011 Salihessen's paper. We just have 7 microbial metabolites recognized for their causal contribution to cardiovascular disease, and 4 out of 7 for their causal role in cardiovascular aging, while we have more than 5,000 microbial metabolites, and we don't know anything about more than 90% of them. And we need to keep in mind that in our intestine, we have 30 to 100 trillions of bacteria, 30 to 10 times more than all eukaryotic cells we have in our body. It's not that wrong if I say we're more bugs than humans. So there's a long road ahead. We need to take gut microbiome science more serious, and they have a causal role. And scientists in the field have such mission, and I personally, as one of the members of this, community, doing, not my share of the science.
That's a really good challenge for the field. So exciting times ahead. So now I pass on to Tatyana.
Cesare, you're working on something a lot of physicians find exciting but scary at the same time. Gene therapy for FH. And we already have statins, PCSK9 inhibitors, and inclisiran. So why do we need gene therapy on top all of that?
Thank you, Tatyana, for the question. So it is true that there are a lot of different therapeutic options, but for specific subpopulations like homozygous FH patients, refractory hypercholesterolemia heterozygous patients, the LDL-C target is still very, very difficult to reach. Moreover, for homozygous FH, this treatment has to be combined together. You start with statins, then you add PCSK9 inhibitors in case of residual activity of the LDL receptor, anti-ANGPTL3, lomitapide, and on top even LDL apheresis for the most severe cases. While gene therapy would change the paradigm. In this case, with a single intravenous administration, in principle you would have the chance to directly arrive to the cause of the disease, restore the protein function that was missing with a long-term effect. And the other advantage would be the in pharmacodynamics context, since all the drugs for now are subjected to peaks and troughs, while gene therapy should result in a maintenance of LDL cholesterol, which is stable over time because of the constitutive expression of the protein that was missing. And indeed, what we proposed in our publication was to deliver a functional copy of the LDL receptor to the hepatocytes by lentiviral vectors, which are integrating vectors. And since they can be maintained during cell division, they could be applied also to pediatric patients where the liver is still growing, which is another advantage. So, yeah.
This is a great point. So, what do you see as the most realistic path to one-shot treatment for FH patient, let's say in the next 5-10 years?
Well, usually people tend to think that safety and efficacy are the main thing to be kept in mind, while actually there are other more important parameters to be kept in mind, like scalability, because once you have proven efficacy in the mouse model or in small animal models, then you move to larger animal models, and sometimes, even in certain cases quite often, for advanced medicinal products like the gene therapy drugs, scalability is the bottleneck that prevents you from reaching the clinic, just because you cannot produce enough drug. So once proven efficacy and safety also in large animals, then the two other pillars of the successful gene therapy to reach the patients have to be kept into account, which are industrial collaborations and patients' engagement. And you have to consider all of these parameters very early in your preclinical studies, because especially if you are an academic, at a certain point, you will have to find an industrial collaborator which will fund the clinical trial. and the patient engagement as well has to come very early, because even if you have the perfect drug, but the people that will benefit from it are not aware of the existence of your therapy, you have to educate them with the pros and also the possible cons of the drug that you are proposing. So in our case...
I think it will be definitely interesting to see. And I have one more question. So your recent study highlights how long-term follow-up can completely change interpretation of a therapy. Do you think the field sometimes moves too quickly towards translation before we understand long-term biological consequences?
Yes, sometimes this unfortunately happens. In our case, we started by keeping the animals alive for 6 months. We saw a full efficacy in terms of LDL cholesterol normalization, completely prevention of atherosclerosis, and the liver tissue was clean and fine. But then we kept the mice for 1 year, which is quite long term in case of a mouse study, since it covers most of the lifespan of the mouse, and since we treated them as juvenile, we captured even hepatocyte proliferation and turnover. And it was at that time where we found the issue, because we observed the toxicity very, very long term at 1 year. So this prompted us to go back to the vector design. So we reduced the potency of expression of the receptor, and we start all over again. And in this case, after 1 year, the safety was reached. But yes, the long term was essential, especially for gene therapy and advanced products. Now regulatory agencies are asking for really long term follow-up, which is good. So I think that in general, this concept is quite shared, and yeah.
Thank you. Pelin, do you have a question for Dr. Kurt?
Berkan, you're a cardiology resident and a clinician-scientist. You see patients and run experiments. A lot of your work is on inflammation, high-sensitivity CRP, interleukin-6, and the new emerging biomarkers in heart failure. With everything we've learned from the CANTOS and the colchicine trials, do you think inflammation has finally earned its seat at the table in routine cardiology practice?
Pelin, thank you for the great question. I would maybe even rephrase the question, because I think it's already sitting at the table. So we knew since decades by now that inflammation, from preclinical studies, but also from clinical studies, that inflammation has a crucial role in the development of cardiovascular disease. And I think what's important here to be considered is that inflammation or inflammatory residual risk is not against but concomitant with other risk factors. So it's framed by Dr. Libby, for example, as a false dichotomy if we think of either lipid risk or inflammatory-driven risk. So in the end, I think it's already sitting at the table, but we as clinicians, we are still a bit hesitant. Our guidelines are hesitant, and in clinical routine, we are hesitant to, for example, consider residual inflammatory risk assessed by high-sensitivity C-reactive protein as a relevant dimension. So the American and the European guidelines consider CRP, hsCRP, as a risk-modifying factor. But we have strong data showing that universal screening of CRP next to LDL cholesterol and Lp(a) could actually help more strongly and more correctly stratify the risk of our patient. And this was exactly where our work that we were able to show last year started at, because we wanted to clean up some myths that were around hsCRP. For example, that it is not a good performer in comparison to other biomarkers, or that it doesn't have a good added value in clinical routine risk stratification. And we were able to assess hsCRP levels in approximately 500,000 patients, and we're able to show that these myths that have that were around in the last couple of years are not really based on strong foundation. And us and other groups as well have shown in various studies in the last years that CRP actually is already sitting at the table.
So adoption is already underway in the clinic. You've also worked on PLIN2 and other less well-known biomarkers in critically ill patients. How do you decide what's worth chasing? What separates a nice signal from something that could actually change practice?
I think this is a very important question in biomarker research and something we will all get asked a lot. I think there are many dimensions to this question. I think first, of course, every biomarker that we want to investigate needs to have a biological basis. And for example, for CRP, we know it's a downstream biomarker. It's active after activation of the NLRP3 inflammasome and the IL-1, IL-6 axis. IL-6 activates the acute phase response in the liver, which then leads to CRP secretion. This is what we can measure with 8 high-sensitivity assays. Also, we know that, for example, IL-6 is causally associated with ASCVD. But for clinical routine, we have CRP as the gold standard biomarker of residual inflammation. And here we have a biomarker with a strong biological foundation, even though it is maybe not causally associated with CVD in comparison to IL-6. But we have a biomarker that works. And I think the other dimensions that are worth considering in biomarker research is that biomarkers need to survive stress tests. So we need to show data. We are obliged to provide evidence that these biomarkers are actually stable. We need to show that after adjustment for various risk factors, these associations remain significant. So we really have to probe these biomarkers to see if they're actually if the signal is consistent. And lastly, I think, and which is the most important, we need to show an added value for clinical risk stratification. So we need to show and we need to provide evidence that when clinicians actually use these biomarkers, for example, hsCRP, which is widely available in clinical routine already, and is already measured all around the world every day, that we can actually change our therapeutic and preventive strategies. And this is, I think, this makes hsCRP, since it really fulfills all these 3 pillars, a really good candidate to be considered for this.
That's a really good framework for decision making. We keep identifying new inflammatory and metabolic biomarkers linked to cardiovascular risk, but clinicians already face information overload. How do you see these markers being integrated into practice in a way that's actually useful and not just more complexity?
Yeah, I think the job of a biomarker should basically be making our lives easier and not more complicated, right? So when we measure a biomarker or any sort of risk stratification tool, it shouldn't make it harder. So we have plenty of tools. We have polygenic risk scores, we have biomarkers, we have other clinical variables that can be assessed. But in the end, we need to have a biomarker that is easily available and make our lives easier and not more complicated. Of course, the future is very promising when we think about automatic integration of risk stratification tools. For example, when we measure biomarkers, why don't we have tools that automatically show us the risk of our patients in clinical routine? I think for this, for example, we need more refined risk stratification score models. For example, we have the score 2 in our clinical routine and primary prevention of ASCVD, but score 2, for example, doesn't really consider the dimension of residual inflammatory risk. So hsCRP in the official model is not integrated. And in our study, for example, we were able to show that hsCRP, when added to the score 2, actually provides incremental value in risk prediction. So this, for example, could make our lives a bit easier in the future. But also, we have a lot of opportunities to enhance risk stratification in the future. And I think we need to put them to a test and in the end see which, on the one hand, is the most feasible, but on the other hand, also brings the greatest added value in this context.
That's the key issue, isn't it? It's the usability in clinic. So now I pass on to Tatyana.
We have a tradition on Athero Talk to ask our guest about a project or an experiment or an idea which didn't work the way you hoped, but ended up shaping how you think today. So did you have this experience?
Yes, I can make the example of the project of our publication. Actually, at the beginning, we were trying to produce our drug, which is usually instructing a cell line to secrete your vector. And actually, the yield was so low that we couldn't even inject that in mice, coming back to the scalability potential issues. And the problem was that the producer cells were overexpressing the receptor that we were empackaging in the lentiviral vector. And since the receptor is one of the receptors that the lentiviral vector itself uses to transduce the cells, the producer cells were capturing back all the drug that they were just producing. And so in the supernatant, we basically got almost anything. And so instead of fighting against biology, I tried to learn from the hypercholesterolemia biology. And so I delivered PCSK9 into the producer cells in order to hope that I would have achieved the degradation of the receptor instead of its exposure on the membrane of producer cells. And indeed it worked. And at that point, the cells were releasing a lot of drug in the supernatant, and we were able to solve the productivity issue. So learn from biology and not fight against it.
Okay, so I think that in general about the difficulties and complexities of the field that I'm working on, I would say that I just remember what I said a little bit earlier. The field is really complex, and what we have is trillions of bacteria, and among these, we don't know anything about even their names. That's one thing, and another difficulty is when we do sequencing, we have millions of gigabytes data that we need to analyze them somehow. Another thing is that we were facing technical limitations over the past decades, especially until early 21st century, when shotgun metagenomics and 16S sequencing came in to support us and scientists in the field to go more in depth at the species, strain levels, and see what's there. But the problem is that in the field, still more than 90% of research and the vast majority of scientists are only working to answer one old-fashioned question, which is what's there? What bacteria are living in intestine? And just to see the association between their abundance and incidence of cardiovascular disease. But the modern fashion question is, what do they do? That's a paradigm shift, and it's time to think about the bacteria's function. What do they produce, and how they can causally be tested. For testing this causality, again, there are so many difficulties because what we really work on are mice. And when we want to translate our findings to humans, we need to think about so many other confounding parameters that affect the gut microbiota diversity, composition. For instance, when we have mice for our experimental setup, we're just giving them a specific diet, CHO diet, or some special ones, high-fat diet, high-cholesterol diet, and they're receiving the same every time a day, while humans intake so many different things during the day. And even though we want to use humanized diet in mice, we still cannot make diversity in that diet. Okay, humanized diet, but every seconds they're available for them. While from morning to night, we see different things in breakfast, lunch, dinner. So that's one of the complexities. Another thing is that in humans, we are receiving so many different medications, from antibiotics to medicines for so many other human diseases, and this can make the translation from mice to humans more difficult. And another thing is that the similarity between the composition of the gut microbiome in mice rather than humans is very less, only 3%. While we need to go more in depth with some new techniques that nowadays are available and we're using them to focus on specific homologs and enzymes that can produce such similar metabolites. And that's exactly the important point that I always say, that this is the paradigm shift from old to modern fashion. I would say these things for now, but yeah, I'm open for further questions.
Thank you for the question. I think asking for ideas, maybe I can continue shortly. I'll try to keep it short. Asking for ideas that actually went wrong is really refreshing, because I think we need to be more transparent about that we all face failures and all face setbacks. And I think what's really important in this context is that we need a good system around us. So good people, a good mentor, I think is really important to really give you an overview about the long run and the long strategy. So as an inflammation researcher, we face systematic problems. So we know that inflammation has been around forever, and we have evidence that have really supported the inflammatory hypothesis with the CANTOS trial as a breakthrough trial in the past, but we still are hesitant with implementation. So this is maybe the overall systematic burden that we have, that we carry around. But on the other hand, of course, individually, we also face setbacks. Since inflammation research is, especially in the last couple of years, such a fast-paced scientific area, you really see so many great people with great ideas. And what's really interesting, and I think what really brings this area forward, is collaboration. So I think in the end, if we collaborate strongly in these area and these fields that are so fast-paced, we can really get these research fields, especially in inflammation, forward as fast as possible. And I think this really helps us all in the end.
It's quite an honest look at daily practice, and I fully agree. We can learn a lot from these experiences. Pelin, do you have more questions?
So one final round to each of you. If there's a young person in this audience who wants to do what you do, what is one piece of advice you'd give them? And if they want to reach out to you, how can they do this?
Difficult question. What I can say that since the field is really complex and complicated, first of all, be patient. Be transparent and never give up. These are very general advices, I can say. But in practice, they really work, because you may run some experiments. I'm just talking about my own field. When you spend months of time for running animal studies, because of the complexity and variability and diversity of the organ that they're working on, which are bacteria, that our control on them are really narrow. So we need to be patient, because it can happen that in one night you see that animals and their bacteria change different reaction to your treatments, to such environmental stressors. So this I can say. And another thing is that try to learn more and more the techniques which are high throughput and progressing. Especially in the field of microbiome science, we are really dependent on techniques. And one of the major reasons that our knowledge about this field was really limited for about a century, since 1902, that Eli Meshnikov for the first time realized that there is a causal connection between gut microbes and longevity and aging, the field was silenced for about a century because of the technical limitations. So learning new techniques, AI would be really important in this case, especially for establishment of microbial age metrics and the microbial signatures for prediction of the incidence of cardiovascular diseases. Yes, this is a package, and I would say like this.
And how can they reach you if they want to? Are you happy to be emailed, or do you have?
Of course. I'm active on social media, on LinkedIn, Twitter. My email address is available. My lab is located at Center for Translational Experimental Cardiology, University Hospital, and University of Zurich. And I can also say my email address, soheil.saeedi@usadish.ch. Don't hesitate to contact me.
Thank you. What can I add after all these great suggestions? I will focus on my field, which is gene therapy. It's a very competitive field, but it's very intriguing. And so since you are working on the drugs of tomorrow, it's very, very exciting. What I can add is try to question yourself and study a lot the disease or the biological process that you are working on. Try to be very, very informed, and then just at that point, try to think out of the box, because you may come out with an idea that may change and solve a present problem in the perspective and in the way that these kind of diseases are treated by now. You can find me on LinkedIn as well, or by email. Thank you.
So I'm happy to add maybe the perspective of a clinician to in the end. And I think I partly even replied to this question in my last comment. First and foremost, I think it is very important to be hardworking, and we really need to focus sometimes. And there are so many distractions in the world, and I think this is something really important. The second part would be that it is really important to find a good mentor. You need to find someone who you look up to, and you need to find someone who you can work with. And I think these are two really important key factors for everyone who is doing research in our area. I think as clinicians, we have the specific role that we, especially in Europe, focus on clinical work, but also do research. But I wouldn't see this as a negative aspect, but more as an opportunity to really translate our findings from research into our daily clinical routine. And this even makes it more motivating to dive in deeper into our findings, because we can really grasp what is going on, and we can really translate this. I think these are two really important points. And I'm really, really happy if anyone wants to reach out, either for just a conversation, or for connection, or for research collaborations. I'm also active on social media, on LinkedIn, on X, but also recommend ResearchGate for everyone. And you can find me online if anyone's interested to get in touch.
Thank you very much for those very valuable pieces of advice. So before we close, we'd love to hear from you. So if anyone has any questions in the audience, please put your hand up, then I can pass on the mic to you.
So Berkan, you actually mentioned mentors. That's actually a question I have for all of you. Can you mention, or say a name of a mentor that has actually been very influential for you guys, and what have you learned from them, or a sentence that has actually pushed you to do something?
So I maybe reply, since I kept the microphone from the last question. Thank you for the question. And I think it's important to find these people around you. For me, very influential people were my PI from my group, Florian Kahles, but also, of course, the chief of my department, Nicolas Marx, who has been really influential for me. But I think it's also important to look up to the people from your research field, to see the people that came came before you that worked on the area for, of course, in inflammation research. Those are Paul Rittger, Peter Libby, Giovanna Luzzo, and many, many more that have shaped the field of cardiovascular inflammation in the last decades. And I think these are very important people to look up to, and everyone has to find their own and follow them. And I think also conferences like this, like the EAS conference where we are currently, are a great opportunity to really get in touch with these people, to see them live, to maybe find them for a short conversation. I think also the EAS Young Fellows program offers a great opportunity to dive in deeper into these potential connections with mentors for the future.
Thank you for your question. So I was quite lucky because the mentor that I am discussing now is Dr. Alessio Cantore, who was my supervisor of the PhD. And he was great since the beginning, because when I was just at the first year of PhD, he was treating me at the same level and was asking me always feedback on my, and also about a lot of other projects. And when we started working on hypercholesterolemia field, again, he trusted me, and he just suggested me what we could do from the gene therapy perspective. And this is the person that I can mention.
Thanks for the question. To be honest, I personally try to learn from people around, not only mentors. That's been my habit always during my life. But I've been always lucky to be surrounded by giants, good people, good mentors. If I would say one name, I say Thomas Michel, Prof. Thomas Michel, my first postdoc mentor in Harvard. Not only, career-wisely, but also personally, he was really friendly, supportive, and lovely person. And I always try to keep my connection to him. And we are now really good friends. We're collaborating. But I learned a lot from him. But still, influential people in my career, I was never in touch with Professor Stanley Hazen in Cleveland Clinic, but his work was really inspiring to me. And that was exactly the first spark for my research about 7-8 years ago in microbiome science field. These are the main two names that I can say now. Thank you.
So if you don't have another question, so we can close.
Yep.
Dear Soheil, Cesare, and Berkan, thank you so much for being here today. It was a great pleasure to talk with you. And I think that this session shows something very important, that no single approach can solve atherosclerosis on its own. And to find solution, we need to pull together basic scientists, translation researchers, and clinicians. To everyone, thank you so much to joining us at the stage. And please follow Outer Talk for more sessions like this. And don't forget to grab our speakers afterwards if you have more questions.